So welcome everybody to the next iteration of the IITE seminar series. It is my great pleasure to welcome Rachel Germain, who is coming from the University of British Columbia and who is going to be continuing the theme that we started last week of talking about theory with an empirical twist. So without further ado, please, Rachel, the floor is yours. As usual, unless you're Rachel, please turn off your microphones for the duration of the talk. Thank you.
Great. Well, thank you everyone for coming, and thank you so much for this invitation. I love that you guys have this seminar series, so I was very excited to be able to present in it. My name is Rachel Germain. I'm an assistant professor at the University of British Columbia since 2019. Today, I am particularly excited to talk to you because I am an empiricist. And although I self-identify as an empiricist, I will say I really like to dabble in verbal theory. Whether or not that makes me a theoretician is debatable. But I am an empiricist who uses theory in a variety of different ways and applications in my work. Before we get into that, just to give you a kind of broad idea of the kinds of stuff we do all kinds of empirical experiments in the lab, in the greenhouse, in the field. We've been working with annual plants, entire communities of plants, more recently actually flower beetles, completely different system, aphids, all kinds of things. And so usually I'm using these systems to understand how species coexist, the basics of community assembly, metapopulations, things like character displacement, evolutionary ecology, speciation. So I have a whole bunch of different experiments where we've been trying to think about how we can do things like parameterized models and things like that to pull out ecological processes that we're interested in. And of course, science operates as a feedback. All of us, to some degree, whether or not we're theoreticians or we're doing experiments, are kind of operating within this framework where essentially experiments are based on hypotheses that stem from theories, whether or not those are mathematical or verbal theories. And those theories are then used to refine experiments. Those experiments test how things might operate in the natural world. They might generate some unexpected findings and in turn revise theory. And what I want to emphasize here is that all of us use theory in our work to some degree. And I'm going to pull out some specific things that I think are kind of unique applications that I've been using in my work and some other things that might be relatable. And so really for the theoretical audience, I think it's helpful to know how empiricists might be using some of the theories that we're driving. And on the empirical side, maybe some of the examples I'll show will give people some ideas on how they could apply these things in their own empirical research. Yeah. And so the purpose of this talk is to really examine theory in service of experiments. And to do that, I'm going to be telling you about 3 empirical experiments. The first is, I'll call this vignette 1, is to think about how theoretical applications have helped understand this concept of environmental filtering and its metaphor. The second vignette is to help our understanding of phylogenetic community assembly. And lastly, I'll be talking about an experiment where we're essentially trying to understand how populations are thriving in variable environments. And in all of these cases, I'll be explicitly pointing out those feedbacks between theory and experiments with a lot more of an emphasis on methods than I would traditionally do in a talk, just to kind of make some of those points. I'm also going to pull in a secret extra section if we have time. I know the purpose of this seminar series is to have more time for discussion, so I'm aiming for about 40 minutes. So we'll see if we can get that in there. And then lastly, I will be discussing some barriers to learning and communicating mathematical theory and how we might be able to overcome those barriers. So to start with vignette 1, which is about the environmental filtering metaphor, the key feedback between theory and experiments that I want to highlight here is that theory can be used to guide experimental design in a few ways. So we're going to start with the concept of what the filtering metaphor is in ecology. It's something that I'm sure is familiar to all of you. We teach it in our undergraduate classes. I know in my second year fundamentals of ecology course, I initially was teaching this idea to them and since have changed that based on some of the findings from this project. But it's the idea that the species that we find distributed in local communities are some non-random subset possibly from the regional pool of species. And we think about that local pool of species arriving through a series of successive filters, the first being an environmental filter that basically selects for species that have particular traits to deal with the abiotic context in an environment, and then some competitive filter that then takes species that might be more different from each other and allows them to use different resources, partition them, and coexist with each other. When people look at trait data, for example, in communities, and they find something like this, where species are really similar to some degree, maybe they have a blue trait, which is helpful in this blue environment, they might think that this is evidence of a strong environmental filter. And maybe there's some competition happening as well, because those blue traits are as differently blue as you can be. The problem with this, of course, is that environmental filtering is not likely independent of competition. So essentially, if we were thinking about the environmental filtering metaphor sensu stricto, in the strictest sense, we might think that it's a species that can live in one environment, this gray environment, but if it were to added to some other environment, this white environment, it would be unable to persist there at all. The problem with this, of course, is that competitive exclusion can lead to a very similar pattern. So here we have a case where this species would be able to live in this other patch if it were able to get there, but purely because there's these other competitors that like that patch, it's actually unable to establish. And so there's this kind of confounding effect of the environment and competition, which the model says should be separate parameters, of course. And of course, in nature, competitors are everywhere. And so to the idea that species are just, dispersing or trying to persist in environments that are free of other species is fairly unrealistic. And this more competitive exclusion scenario is the more realistic scenario. Unfortunately, about 85% of studies confound competition with environmental filtering. And so my goal here, the question is evidence of environmental filtering consistent in the presence and absence of competitors? And does this answer depend or differ if we contrast among different abiotic environments? If we wanted to actually test this, again, dealing with this idea that the species that are present in communities are only the ones that can persist there, what we want to do is actually put species in those environments where we think they might not be able to be, and actually test if they're unable to persist. Now, how do we test persistence? One way to do it is to do an invasibility experiment. So I'm going to describe what that is and how to interpret it. But essentially, in this case, I want to test two things. I want to test the ability of a population of a species to establish in the absence of competitors. This is what I'm going to refer to as invasion purely in the abiotic environment, in the absence of competitors. So this E is denoting that. And also the ability of a population of species to invade when another species is present. By actually looking at the population, per capita population growth rates under these two scenarios, we can ask, are these populations of species predicted to fail to persist or having an invasion threshold below 1? Or are they predicted to persist or that invasion threshold above 1? And here I've done this in a replicate experiment with two soil moisture environments, a wet and a dry environment, and I've done this across 30 different pairs of species. Now, importantly, you might think that you might just be able to test this invasion scenario when that one species is being invaded among some random background of some other species at some high density or something like that. But invasion experiment is actually a bit more precise than that. In order to estimate invasibility, essentially what we want to do is to take one species and allow it to reach its equilibrium population size. And at that point, we try to invade the second species. When we invade that second species at a low density, when its competitor is at its equilibrium, we're essentially asking, can that invading species persist and invade that community? And so we're looking for a positive growth rate here. And we can do this for both species reciprocally to say, like, okay, is this a coexistence scenario? Is this a case where we have competitive dominance of one species or another? And this is great. These invasibility experiments are super useful. They kind of simulate what would happen if a species was entering a new environment that it wasn't in previously, or what happens if it's driven to low density, if it has some ability to buffer its own growth and reinvade. But I work with annual plants. And in my case, each time step is essentially 1 year. So if I wanted to do something like this, I just don't have this kind of time. It would be 50 years of work, I'd say. And so an empirical modification that I use is space for time substitutions. So I want to walk you through what a type of experiment with a space for time substitution might look like to get at something like invasibility. And so let's break down what invisibility is in the context of a population model like this one. So here, if we're considering the invisibility of one species, species I, when it's invading into a new environment, theoretically what that means is that that species is absent when it's entering that environment. And so we can kind of disregard the intraspecific component. It's not impacting itself, it's theoretically absent. And again, we're invading it when the other competitor is at its equilibrium density. The equilibrium density, of course, we know exactly how to calculate that as well, because it's just the single species equilibrium of that other species. We can then, of course, do this reciprocally. So we can look at the invasion of both species when the other one is at its equilibrium density. To actually parameterize a model like this, we set up experiments where we modify the initial density of competitors, and we might look at something like the per capita growth of each species as you change that density of competitors. So you might get some sort of growth response like this. By looking at the intercept of these per capita population growth rates, essentially when there are no competitors present, this is what we can use when we fit our models to identify something like an intrinsic growth rate. And then something like the sensitivity of these population growth rates to, for example, the density of intraspecific competitors can give us something like our per capita interaction coefficient. And of course, we can do the same thing by modifying the density of interspecific competitors to get at the per capita interspecific competition coefficient. And then do this for the other species, where we're tracking now the per capita population growth rate of species J. And what's great about this is you'll now notice we have all of the ingredients that we need to kind of plug everything into these invisibility equations and actually ask whether or not those species can invade each other, if one's going to be dominant, if some are going to be excluded, whatnot. So to get to the findings from this experiment, so again, to remind you, the purpose here was to identify whether or not the environmental filtering and competition are kind of acting independently. But here we actually find that environmental filtering only emerges with competition. So I'm going to walk you through some slides. They're kind of complicated, but I'm going to kind of walk you through this. But again, first, we're dealing with the invasion growth rates in the absence of competitors. So it's basically like, can you live in this abiotic environment or not? And again, we have our persistence threshold. So above this threshold, they're predicted to persist, and below it, they're going to be excluded. Each of these dots is a different species, and they're grown in two different environments. So each line connecting a series of dots is how any one species is doing in a dry or wet environment. The first thing that I want you to notice here is that most species can persist in the absence of competitors. That's not terribly surprising. This is done in a greenhouse environment. It's pretty cushy. You'll also notice that there aren't a lot of very dramatic slopes to some of these relationships. So even though we have a wet and a dry environment, it's overall pretty decent. And in fact, if we were to look at the number of species that fall below that persistence threshold, we find that it's only one case of that in a dry environment. Now where it gets interesting, if we compare these findings to the invasion growth rates when they're reared with competitors, and we've done those invasibility experiments, now we can see that the answer is quite different. Basically everything, all of those growth rates are pushed way down, some to a greater extent than others, is the first thing. And now you can also see that there's some cases where a species can persist in one environment with that competitor, but not in another environment. So there's kind of like filtering that's happening at the scale of different soil moisture environments. So now tons of environmental filtering and the abiotic environment is actually starting to, those differences between the abiotic environment are starting to matter. And so this is telling us what's happening here is that competition is affecting the strength of filtering, and also the environment itself is affecting that strength of competition. And that's why you have that differential success across environments. I think it helps to break this down by a specific pair of species to better understand what's happening. So here we're comparing two Licinia species, Licinia californica and Licinia glabrata. Again, first we'll look at their invasion growth rates in the absence of each other. So when they're purely responding to the abiotic environment, you can see they both do super well, not a huge effect of soil moisture environment, basically no effect. And they're, one's slightly better than the other, but it's not very dramatic. When we look at them instead, when they are actually competing with each other across these two environments, you can see that there's one case where Glabrata, for example, does much better, is competitively dominant in a wet environment, and actually dominates and excludes the Californica species. So here we have a case where in one soil moisture environment, these two species are actually able to coexist with each other as predicted by observation growth rates. And in another environment, we actually have competitive exclusion and dominance. And so this is a question raised in a tree paper by Carolyn Tucker and Mark Cadot and some other authors, asking, should the environmental filtering metaphor be abandoned? A response based on our data is that yes, most environments contain competitors, and competitors strongly modify persistence. So maybe this is kind of like an older way of thinking about things. Or alternatively, you say no, maybe we're asking the wrong question. This is really revealing this interesting interplay between species realized and fundamental niches. And instead of thinking about things as a hierarchy, maybe we should be coming at it from a process-based interaction up type perspective. But importantly, in the context of this talk, this is a case where theory has helped guide what we measure, in this case, population growth rates, what conditions to measure it in, how we set up those different density trials, for example, and also how to analyze this data by taking those estimated parameters from model fitting and actually knowing exactly how to apply the math to estimate something like invasibility and invasion growth rates. And one other thing that I'll say is, I see this experiment as somewhat of a proof of concept that happened in a greenhouse, again, where things are pretty cushy environments. But we are testing this right now with this pretty massive experiment in California, where we have 18 different sites all over the place, and we're doing these invasion trials with and without competitors to kind of disentangle these things in the field. And this is a student of mine, Megan Stoika, who's now in Lauren Shoemaker's lab, and we're all kind of working on this together, which is exciting. Okay. The second vignette, we're going to be talking about phylogenetic DNA assembly. This is a case where I think the interplay between theory and experiments here is this ability to settle a debate by testing some assumptions. I'm going to start off with a gratuitous Darwin quote, where he said, as closely related species have some similarity in their habits and structure, the struggle will generally be more severe between closely related species. And what he meant by that is if we have a community of species that have some sort of phylogenetic relationship, we have some species that are more similar than others, both phylogenetically and with traits, then we might expect this non-random exclusion of closely related species, such that when we look at communities that are competitive, we would expect that those communities are composed of species that are more distantly related than expected by chance. And this was an expectation of tons and tons and tons of work that happened, at the time when I did this project. It was, I'd say this was the primary thing you would encounter in a journal at the time. And it's this concept that we've been referring to as phylogenetic limiting similarity. You're just too similar phylogenetically to live together. And this it was often taken as an assumption of many papers. We're kind of saying that we know that closely related species are similar to each other, therefore we would expect this pattern. But that assumption itself was rarely tested at the time. Of course, ecological theory tells us that not all differences among species promote coexistence via competition. So first we'll talk about those that actually do promote coexistence. Here we have a case where we have two species that are different from each other in where they're accessing resources in the soil profile. You can imagine that this kind of difference would stabilize coexistence because basically it's allowing them to use and access different resources and partition their environments. So in that case, differences are good. They help coexistence. The other one that was kind of left behind at the time are those differences that actually preclude coexistence. So here again, we have two species. In this case, they're actually using resources at the same depth in the soil profile, but one is just better at it. It has more roots, maybe the root structure is a certain way or something. And so those are differences that actually would cause a competitive hierarchy that would lead the inferior competitor, all else being equal, to potentially go extinct in this environment. And these are what people have been referring to in the ecological literature as a fitness difference, not to be confused with evolutionary fitness, but this is kind of a competitive ability difference is the way to think about this. Now, the trick with this is that coexistence ultimately depends on the relative evolutionary rates of stabilizing and fitness differences, if those assumptions of phylogenetic limiting similarity are correct. And so here we have a coexistence metric, which is essentially the relative strength of those stabilizing and destabilizing differences. If we're starting at a phylogenetic distance of 0, and those stabilizing differences do in fact evolve faster than fitness differences, then yes, we would expect closely related species to coexist less often than expected by chance. By contrast, if those fitness differences evolve faster, we kind of end up in this exclusion zone, where close relatives are actually more likely than distant relatives to coexist with each other. The problem is, at the time, we had no idea how these two rates evolved relative to one another, which is a problem because, again, a lot of work at the time was based on this assumption implicitly that stabilizing differences do, in fact, evolve faster. Another really fascinating, I think, complication that's really interesting here is that the evolution of these two types of differences likely depend on biogeographic history. And so we're going to talk about stabilizing differences first, but the idea here is we're looking at a range of phylogenetic distances from very closely related to very distantly. And stabilizing differences are something that are bound between 0 and 1. So 0 means that they're basically ecologically identical, and 1 means that they're very dissimilar and they're partitioning their environments. Under something like character displacement, which is a very well-studied phenomenon in evolutionary biology, you would think that species that share a place in space or occur sympatrically or overlap biogeographically would be experiencing pressure to be different. So that's kind of the expectation. It kind of pushes these species to use resources differently, and that's a stabilizing thing. So you'd expect stabilizing differences to evolve at a faster rate in those sympatric populations that have been, or species that have been overlapping through time, and you wouldn't expect that for things that have occurred allopatrically. By contrast, this is idea that species that do occur sympatrically can only be so different before they go extinct, and they're actually at the same time as this evolutionary pressure for similarities. And so we might think that in a sympatric case, that these fitness differences between species should actually be minimal over evolutionary time compared to allopatry, where they don't have that history of trying to keep up with each other. So different possible effects of biogeographic history on these two types of ecological differences. To that sense, I did a big greenhouse experiment where I had species that were either native to California or Spain. I set up things in pairwise competition experiments, specifically choosing those pairs so that the species that I chose did not have overlapping branch lengths, which is kind of an interesting feature. It's kind of like an experimental phylogenetic independent contrast. And then essentially I fit models to data in the same kind of experiment that I described earlier to parameterize that Beverton-Holt model and estimate those stabilizing differences and those fitness differences and how they come together to predict whether or not a pair of species are likely to coexist or not. And I did this for 20 pairs of competing species. So that's our level of replication. It's a species pair. We have 10 sympatric California species and 10 allopatric California Spain species. Okay, so this is set up exactly as the prediction figures that I showed you earlier. Just to remind you of what the expectations are, we're going to start with stabilizing differences. So again, I said that under character displacement, you'd expect species that overlap biogeographically would experience this pressure for differences over evolutionary time. So you'd expect this kind of rapid increase in stabilizing differences. So you get more and more dissimilar. And interestingly, that's exactly what we see. And in fact, if you kind of look at, again, this is bound between 0 and 1. If we look at about 90 million years of ecological separation, we're kind of becoming as different as we possibly can be. So it's kind of like saturating kind of relationship. If we compare this instead to species that have no history of occurring sympatrically or they're completely different biogeographic regions, there's just no relationship at all. It's a complete shotgun. So having this contrast, this biogeographic contrast, gives us a bit of confidence in the mechanism underlying this difference. If instead we look at fitness differences now, again, we expect under things like the Red Queen hypothesis that sympatric populations would evolve fitness differences at a slower rate than they do in allopatric populations. Again, that's exactly what we find. So the slope of the relationship for the sympatric pairs is shallower than the allopatric pairs. You might have noticed, however, that I expected this kind of saturating relationship, that maybe as you get older and older and more separate, that maybe the pressure would kind of slow down for these types of differences. For example, if you're using niches differently. But you might have noticed that this is on a log scale. And what this is showing actually is that this is more of an accelerating relationship. And we haven't yet found like the maximum amount of differences between species before this starts to level out, which I think is kind of an interesting nuance of this result. And of course, I told you that coexistence is kind of the net effect of these stabilizing effects that help coexistence and these fitness differences that preclude coexistence. And so because I showed you that these sympatric populations evolve those stabilizing differences over time, they're actually evolving at the exact same rate as those fitness differences that actually hurt coexistence. And so that means that if we look at the net effect of this for coexistence, there's really no relationship between phylogenetic distance and your probability of coexisting. By contrast, if you look at those allopatric populations, those fitness differences are actually evolving at a much faster rate than those stabilizing differences that have no evolutionary signal. Because of that, distant relatives are actually less likely to coexist with each other. And so the main thing here is that it doesn't matter if you're sympatric or allopatric. Close relatives are basically, there's no probability, or the close relatives are not competing more strongly than distant relatives, in contrast to this assumption and expectation that's been long held. And so Darwin got this one wrong. One other thing I'd like to point out about this, which is kind of cool, is that historical contingencies ended up being a really important feature of these results. There's this really clear role of biogeographic history in shaping the evolution of these present-day competitive interactions. And in fact, if we had ignored this history and instead treated all pairs equal, this type of historical contingency would actually obscure those evolutionary relationships. So if I kind of remove all of the coloring of those points, you can see it looks more like a kind of shotgun approach. So very important to pull those out. And I think, again, the macroevolutionary consequences of that are pretty interesting. A few research projects that I have on similar themes right now, they're in various stages of being in revisions and published, is I have a project on character displacement and how that plays out on microevolutionary timescales, projects looking at the evolution of demographic trade-offs when you have species that compete with each other, and also things like local adaptation in a community context where we fit interaction parameters between focal species and all of the other species it interacts with in communities, and see interesting things like the Lee effects and stuff that you wouldn't pull out necessarily in a greenhouse experiment. Okay, great. So the last project I want to tell you about is, I think I wanted to talk about this one because it's a very different application of theory, I would say. It wasn't used to design the experiment, but more to give context for what we found. And this one's on the topic of life in variable environments. Specifically here, I'll use theoretical tools to provide a proof of concept to better understand an unexpected result. And so to set this one up, basically the question here was, how does dormancy evolve in environments with more temporal variability? And this is a project done by my undergraduate student, Kelly Slimman, who's now a PhD student in the Agro Lab at Cornell.
Hi, I'm Kelly.
That's Kelly. And basically the thought behind this experiment is that in California, where I work, naturally in the field, there's this really interesting phenomenon where more productive sites actually are more variable through time. They respond way more dramatically to the climate, whereas sites that are very unproductive and harsh are less variable. And so we were kind of using this natural variation to make predictions for how dormancy might evolve in this system. The basic expectation, of course, is that since dormancy is a strategy for coping in temporally variable environments, we might expect to see more dormancy in more variable environments, right? It's pretty basic. In this experiment, we looked at the evolution of dispersal and other things as well. But that just seemed obvious and intuitive, this relationship, right? You'd expect this for obvious reasons. To test this, we set up experimental design where we have a germination assay, where we have 23 populations of a grass species that we've collected from these sites that span that variability gradient. We grew them in a greenhouse for 1 generation to remove any maternal effects and those kinds of things. And then did a germination assay on the offspring in pots containing serpentine soil on the offspring generation. So again, same figure as before. To remind you of what the result or the expected result was here, positive relationship between dormancy rate and environmental variability. Again, intuitive and obvious, right? But we see the complete opposite pattern.
How surprising.
It was very surprising. We did not expect this at all. At first, this made literally no sense to us, I would say. And to be honest, I thought about it for days and days. And then one day it just hit me. I'm like, okay, maybe this is what the answer is. And the answer is that if I think about these sites that span this productivity gradient, this is what they look like. This is one of the harsh sites on the top and the benign sites on the bottom. And you can see in this harsh site, there's rocks, there's bare soil. It's if I were a grass, it's probably pretty crappy, right? If I land in the wrong part of this landscape, I might not germinate, or if I germinate, I might die, right? So the question here is, what if dormancy actually evolves to combat this fine scale spatial heterogeneity? If this were true, I would be looking at sites that either had low productivity or high productivity. I would be looking at the proportion of microsites that are unsuitable to germination. And I would have this prediction that unproductive sites have a higher proportion of microsites that are unsuitable to germination. Or the visual is this: more unsuitable microsites in a low productivity habitat. To test this, I basically went back to these sites, and in a grid, I penetrated the ground to see, if I were a seed that were trying to lay out some roots, would I be able to actually get into this ground at all, or are there rocks and other things that are preventing me from doing this? And in fact, when we do that, that is the case. Which again, kind of makes sense based on what I've observed at that field site. But of course, that's not a direct test, right? In order to test that directly, we need a bunch of sites that varied in how spatially heterogeneous there were, and we would be able to actually isolate that potential mechanism. So what I wanted to do here was actually test the plausibility that dormancy could help combat microsite variation using a simulation, because to my knowledge, nobody's identified this possible mechanism of dormancy. It's usually thought as being something for temporal variation, and here it's a completely different mechanism. It's spatial variation. So what I did here is I set up basically a lottery model with a few differences, an individual-based model, where I've set it so there's no dispersal limitation at all. I've set it such that dormancy needs to be plastic, so that if you end up in a bad site, you stay dormant. If you end up in a good site, you break that dormancy. And the main things that I'm altering here are the proportion of microsites that are unsuitable to germination. So here you can see this landscape is entirely suitable, and also a landscape that's entirely unsuitable, and also varying the dormancy rates of the populations that we're dealing with, and looking at the population size when it reaches equilibrium. Not surprisingly, when there's no dormancy, these populations are unable to persist beyond some threshold of unsuitable microsites. So when it's about 50% unsuitable, and this is just based on the thresholds of fecundity that I've set, but they're basically just unable to persist there. And this is parameterized based on the experimental data as well. But interestingly, when we look at the highest dormancy rate, here they have a 90% probability of staying dormant when they enter a bad site. This really pushes how bad a landscape can be in terms of microsites to still allow persistence. And of course, everything in between if you vary all the different dormancy rates. And so to me, this is really just a proof of concept. I'm not going to say this is a general theory or anything, but just the fact that I could use these simulations to suggest that this mechanism could plausibly happen happen, I think is a really interesting feedback between theory and experiments. I like this project too because I think it comments a little bit on the collapse of space and time in a dormancy context. Around the time when I was working on this project, I'd watched this really excellent movie that I highly recommend called Interstellar, where the tagline is love, which I'll modify to say, and dormant life stages are the one thing that transcends space and time. And what do I mean by that? So if we think about a plant that's in some environment that has patches, again, normally we think of dormancy as being something that allows a seed to persist through time. So now it's in this one patch here with a blue environment at time 1. If the environment changes, it's now experiencing a different environment. We're keeping track of that. Again, the next year, different environment. And you can see there's this time series of environments here that the seed is experiencing as it remains dormant. However, we can imagine something similar happening in space. So we have this seed that's in this one patch, again, it's experiencing it. Let's say it disperses a short distance to the side. It's now experiencing, again, a different environment. If it disperses again, moves a short distance to the side, it's experiencing a very different environment. And this is completely plausible because, in these microsites, for example, within a plot, this is happening on the scale of, say, 10 centimeters. And so the question here is maybe variability in time and space are actually experienced in the same way. So we've got this, yes, black hole, like collapse of space and time, which I find fun. All right, I think I'm going to do it. So we have this secret extra section. It's actually, I won't say that this is a specific project, but it's something that I've been doing a lot recently again, it's the power of using theory and experiments. And it's specifically using simulation to choose between experimental designs. What do I mean by that? So here's a photo of one typical kind of experiment that I would do. Some of these experiments that generate the kind of data that I just showed you are massive. So here's one, for example, we had 900 pots, and the designs are really complicated. We have different densities. We have to collect the seeds of everything. It's just out of control, huge. And it's always this balancing act between the size of the experiment we can reasonably do and how accurate it is. And again, we're trying to parameterize often, like any one parameter in here requires a lot of power in order to get it properly, estimated properly. And there's all kinds of different designs we could do that have different levels of efficiency and accuracy. So here's one, for example, where you might do these trials where you have focal individuals and you're varying intraspecific and interspecific densities in kind of all combinations. You might cleverly find some way to reduce this design. You can see this looks very appealing. It's half the size. Now we have 2 focal individuals that are kind of nested within these different background communities. We might ask, how do we estimate the intrinsic growth rates? We might be able to get it simply from the intercepts of fitting data to these experiments, or we might need to actually have a separate treatment, for example, where we grow things completely in the absence of other individuals. Or again, maybe we can make this slightly more efficient and have 2 focal individuals in a pot, for example. And so, which is best? I know logistically which is easiest, but that doesn't mean I'm going to get, the most accuracy that I need in order to make the conclusions that I want, right? And so what I've been doing recently, I highly recommend this for anyone who's doing these kinds of model fitting exercises, is for each experimental design, these different options that I have, I actually create a fake data set under each experimental design, where I essentially simulate the population sizes that I would expect in the next time step, given realistic noise that might happen in a real experiment. So again, I set up these models. I'll give it some fake initial population densities that reflect, for example, the number of seeds that I would put in a pot. I give it some realistic probability of germinating, which affects how many things are actually going to be interacting, and also set some kind of noise on the seed production that we get out of it, because nature is imperfect, right? I then get this data frame of what I put in, what I get out of this simulated data, and I actually fit models to it just like I would with my own data, like literally the exact same models. And what I can do here is, because I know what parameters I put into this model, I can compare that to what I got out of it, and actually look for discrepancies, okay, like which kind of design, how much replication is needed in order to accurately estimate these parameters that I know have the value that they have. And so I'm not going to walk you through every design is different, right? But this has been hugely helpful for designing field experiments. One interesting discrepancy that I want to point out, I want to point this out because I think it's of empirical and theoretical interest. And that's one problem I encountered when I was doing this model fitting, is that I've been finding that my intrinsic growth rates have been grossly underestimated in some cases, while the intra-specific interaction coefficient has been grossly overestimated. And when you look at the posterior distributions, because I do this with Bayesian model fitting, I found that these two parameters end up being highly correlated with each other, which is indicating that something is wrong. And so I wanted to figure out why. So what's our expectation of what an intrinsic growth rate is, right? Our expectation is that an intrinsic growth rate is basically your ability to produce seeds when you're completely in the absence of competitors. So we're kind of assuming that this denominator goes away and it's entirely just the lambda is kind of everything. The reality is when we have this single individual in an environment and we plug this into our model, we actually have a population size of 1. And this causes the intra-specific coefficient to actually remain and try to be fit in that model. What this is saying is that a singular individual is actually competing with itself, and it's causing this weird fitting between intrinsic growth rates and intra-specific interaction coefficients. If lambda is technically growth at a theoretical population size of 0, it's kind of devised assuming that population size is kind of a continuous density. So that might make sense in continuous models. But in cases where we're dealing with populations of a finite size, there's this empirical problem that can actually lead to misfitting of your data. But also it just seems biologically weird to have this assumption, again, in finite communities where you can't have a fractional individual. And the solution here is super easy. All you have to do is subtract that self individual from itself, and then the fitting works perfectly. But it's just something interesting that I noticed in setting up these simulations that otherwise it wouldn't have occurred to me. Okay, so back to theory in service of experiments. Basically, there's a number of different ways in which theory has actually led me to set up and interpret my experiments in different ways, both the motivation, the experimental design, the analysis, and the inferences. The ones in blue are the ones that I've told you about already. You might want to test an assumption or a prediction. You might set up your experimental conditions to mirror something theoretically. It affects the choice of variables you might pick. If you do these simulations to choose your design, all of these different kinds of things. What I want to say is that, of course, I'm using theory as kind of this way of guiding the experiments that I do. Science doesn't have to be very strongly based on theory in order to be, strong science, but it is something that I personally use in my own work. At the same time, there's this feedback between the empirical results and the theory, and that's where these nuances and these unexpected findings can lead theory in new directions. For example, what happens if dormancy is in fact something to help combat microgeographic variation instead of temporal variability? I'd love to see somebody incorporate that into their model. But interestingly, there's this rift that people have noticed that, the links between theory and empirical work aren't necessarily as strong as they could be in research. And why is that? There's a number of different reasons, but one that I'll point out is that for many of us, and I'll say this has been true for me and is something I'm continuing to work on, is that the mathematical aspects of theory can feel very unapproachable. And so we're looking for solutions there. How can we fix this problem for people like me, empiricists? So that leads me to a shameless plug for a new paper that just came out in MNAT. It's called An Empiricist's Guide to Using Ecological Theory. And it's the product of this really fun working group we did where we essentially brought together a group of empiricists and theoreticians and also graduate students who were currently learning how to do theory. This project was co-led by myself and Tess Granger, who's highlighted here. And this paper is essentially broken into a few different pieces. The first is what is theory and how is it created and how does that parallel how an experiment is done? How can theory be used by empiricists? That's kind of largely mirroring what I talked about today. but also how can one begin to approach the mathematical aspects of theory? How do we interpret certain types of figures that commonly occur in theoretical papers? How do we start to handle equations, for example? And Dan Bolnick said on Twitter, this article should be used in every ecology course everywhere. So yeah, check it out. It's available now. And of course, enhancing or kind of breaking through this barrier between theoretical and empirical ecology, in part, is something that we as empiricists can find ways to kind of break through this barrier. But I do think there's another side to that. And that other side is how theory is communicated when papers are kind of being written and who the intended audience is in mind. And so I will say we do have a follow-up paper that's hopefully going to come out soon in bioscience. It's led by Gilles Enriquez and Will O. That's kind of getting at this issue more from the theoretician's perspective and how, if somebody wanted to better engage with empiricists, how someone might approach that. And so that is, everything. I'm happy to engage in discussion, and thank you for coming.
Thank you very, very much for the both very interesting and very entertaining talk. So now it's time for questions. And I see already that in the chat, Eva Kijdi has put in reference for a paper by Hans Metz and colleagues. And yeah, Eva, please go ahead.
Yes, thank you. Thank you for this thought-provoking talk. And I'm sorry I sent that reference a little too early. By accident, but I got actually quite excited about your germination results because there is this little known paper by Hans Metz and colleagues, which is now in somewhere in the chat, and I also send the journal name and year, etc. So that they offer an alternative model for germination. So it is a very widespread idea that delayed germination is good to cope with temporal variability. But they point out that it's also good to cope with microhabitat variability. So I think it's very close to your idea, even though it's not the same as far as I can remember. But it would be exciting to compare your results with the predictions of this model.
Absolutely. Is it a movement thing? Is it like literally like you can access new microsites?
Mm-hmm.
Awesome. Yeah, I'm going to check that out for sure.
Thank you. Matthew, please go ahead.
Yeah, I have a question again about the life in a variable environment experiment. So thinking about similar situations I've seen, I think 3 questions. Do seeds need to know what environment they're in? Do they benefit if they do? And can they know? And my guess is the answers will be no, yes, and it depends. But I'd be interested to hear what you think.
So more like cues, what kind of cues they get? Is that the question? Yeah, I mean, in these spatially variable ones, so it's all through, I think that germination is very sensitive to those cues. And one reason why I'll say that is because, I alluded to this one experiment that was for a completely different purpose, but it was essentially looking at how density dependent growth is in serpentine environments. So it actually transplanted into the field. And there was this extremely strong elite effect where, it wasn't until we had like 10, 15 individuals in the same neighborhood as another individual that it actually became, viable at all. And that was in a model that incorporated both the germination, the survival, and fecundity. And I'm almost sure that the majority of that is through the germination fraction. And the idea that it's like when you have other individuals around, it kind of gives you this protective barrier that changes the microclimate. And that's just absent in these really harsh patches. Like that photo I showed is just, it's bare ground, rock, they could land on a rock. It's hot in that environment. And yeah, so I think those allele effects that I saw, I would be shocked if it had, although I'm always surprised, but I would be shocked if it wasn't due to that microclimate that's caused by those other individuals. Yeah. And actually, one other thing that I'll say is, in that experiment, we did the dormancy trials both in a harsh environment and in a benign environment, and you don't see that at all in the benign environment. And so what it is that it seems like there's this, it's not dormancy itself that evolved in the harsh versus benign serpentine sites, but it's actually their plasticity or their ability to sense the environment and cue that germination. And in fact, in that model that I showed you, the individual-based model, if you don't make dormancy plastic, it is unable to affect persistence in spatial heterogeneity. You actually need the plastic dormancy in order for it to stabilize the populations. Which is kind of interesting.
Thank you so much. Abdel, you had your hand up. However, in the chat, I see that Liz Pastor had something. I don't know if you wanted to say something.
Yeah.
Yeah, please go ahead.
Yes, I would like to. I enjoyed this very much because it is so difficult to work with plants, and you've made so many very valuable and interesting things. My remark concerns the problem of population regulation. And here would be very, very interesting to see that what they compete for. So what are the checks? What are the regulating variables? How the plant affect each other? And why am I saying that? That when you say that Darwin was wrong, then he might be wrong saying that always competition will be most intense between the close relatives. But Darwin said that, but Darwin gave a reasoning, and that's what I put into they because they have this, they live at the same place, they have the same food, and they have the same enemies. And these are the regulating things which determine whether they can coexist together or not. So my question is that I see that you can always manipulate population density, and by manipulating population density and waiting until the population gets to the equilibrium, then you change those environmental regulating conditions which matters. But the other thing is whether you can identify these regulating conditions. So, yeah.
Yeah, maybe I'll address that in a few ways. So, I mean, of course, the power of these experiments is that it's phenomenological, and so it's kind of like the net effect of all of those things. Of course, in a greenhouse, we remove some of the realism, just because you have to in a greenhouse. For example, we don't have like natural enemies and things like that. But so I mean, I like working with annual plants because it allows you to kind of skip the details, but I'm personally fascinated by those details, and it's something that I've kind of found that I'm unable to do with the plants. And the reason why I say that is, like, I've measured traits in these experiments. I've measured everything you measure. People measure things like leaf length and root depth and all of these things, and I have yet to find a single trait that can predict anything. And like that makes complete sense to me because what we're not measuring are things like uptake rate of specific, compounds or nitrogen or whatnot. What we're not measuring is the metabolic requirements that plants have. And it's very difficult, I'd say, to do that with plants. It is something I'm really excited to do actually with the flower beetles, which I've started working with in the last, actually because of COVID, we switched to flower beetles. But yeah, I mean, basically something with a mouse where you can actually measure its consumption rate. You can see how it takes those consumed resources and convert them into fitness. Those are things that, and with plants, of course, you could do things like add nitrogen or whatever and see how they respond to that, more like R-star kinds of experiments. But I do like the possibilities of trying to get at the mechanisms a lot more clearly.
Yeah. because then it gets natural that those environmental conditions which don't regulate will influence the ones, the levels of the others which regulate, and then the competitors also. So then okay.
Well, it's funny to add on that. People often think about things like temperature or certain abiotic things as being, not finite, and they're not competing for it. So it's like a density independent regulation. But at the same time, individuals still compete for space, right? And so that's the case for, even though it's something you might think is density independent, I do feel like there are density dependent influences of those things.
Yeah, I agree. That would be my starting assumption as well. Thank you very much.
Thanks so much. Unfortunately, Abda has left in the meantime, but Jonathan, please go ahead.
Right. Thank you, Rachel, for that really interesting talk. I know a lot of it, so I'm not going to ask about the stuff I already know, but I will ask you about the last section where you have the dormancy evolution. And I guess I have two questions that are related. One is, I'm assuming that in your simulation model, when a seed doesn't germinate, it has a chance to move to a different site in a future year.
Yes,
Okay.
it does.
Good. But the other thing I was going to say was, another alternative explanation for what you're seeing is that it's not the variability per se that selects for dormancy. It's the probability of having a year in which your lifetime reproductive success is less than 1, right? So even if I fluctuate from 1 to a billion, I'll have no dormancy whatsoever. So I guess my question was, is it possible that these sites on the serpentine hummocks that you're looking at are harsher, such that the dormancy may be evolving because there's a greater chance of actually having a year in which, or several years in which germination would lead to actually negative population growth? Whereas in the other sites, even if they're more variable, they're more productive, and there might actually be lower risk of germinating, even if it means you have no idea what your perceived production is going to be.
Yeah, that's a great question. I've been thinking about this a lot, actually, because there's so many arguments either way. But one thing that is true is that in a harsh environment, it's kind of crappy everywhere. When things switch from being a bad climate year to a good climate year, there's often, in the competitive sites, there's this like transition year where like the climate's really good and competition isn't that intense yet. And so they have this chance to blow up. That's in the benign sites, the like good abiotic sites. But then competition kicks in, and it's a very like short window of success. In the harsh sites, in a bad year, it's just bad overall. In a good year, again, there's very few microsites that are actually good, but the few individuals that make it to those sites tend to do really well on average. And so I think that there's this interesting multiplicative thing between how each individual is doing and how big the population is and how those are responding to the climate. So I do agree that that is a plausible alternative mechanism. Here I was really just trying to say, like, it could be either, right? And I never thought about this microgeographic issue, and it wasn't until I saw those results that also seemed plausible. So I'm like, is this possible at all, right? So that's kind of the proof of concept motivation behind that. There are alternative explanations for the field patterns, but is this one of those possibilities that, again, totally non-intuitive unless you think about it? Is it literally plausible at all? So that's the thinking behind that.
Okay, thank you.
Yeah, does that answer your question?
Yeah.
Thanks so much. Giza, please go ahead.
Thank you for the lecture. It was very interesting, and I also took a look on your new paper, and it was very nice also. And from your lecture, I would be more interested in the issue of stabilizing difference versus the fitness difference, where I understand the increasing nature of the stabilizing difference because they just evolve away from the competition of the another one. But I don't understand what is happening with the fitness. What is your underlying model of evolution?
Right. So you mean, why do we have this expectation that the allopatric ones can have more fitness differences than the sympatric ones? Is that the question?
You mean fitness is your lambda, which is the growth rate in the empty environment, right?
So the fitness difference, it's kind of is like the multiplication of the demographic portion of it. So how many more seeds do you produce than somebody else, but also the sensitivity of each of those seeds to competition. So it's kind of like a composite, like the actual sensitivity to like per capita interactions with other species, and also the demographic component. So it's kind of two things.
Okay, but this is the point which I can you say the form? Can you show the formula?
Oh, okay, yeah, sure.
This is the point which I don't understand.
Yeah, let's find that. That is here. Yes.
Yeah.
So here's the lambdas, and then the
yes, and okay. Okay, I don't really understand what it means. It's this is the ratio, okay, because it is too small and I don't really see the indices.
Oh, okay.
But what is your just okay? Right, it's perfect. And what is the justification of having this is as the fitness? Why this is fitness?
Well, first, it's hopefully this isn't, yeah, it's equal. It's basically, I would call it a competitive ability difference, but the idea is that there's two ways to win. One way to win is just producing way more offspring, right? And
yes.
all else being equal, that would make you win, right? But if you're producing, for example, a ton of offspring that suck, for example, that's not going to help you. That actually could balance out that demographic advantage that you have. And by sucking, I mean the second part of the equation. This is like how much you suck, or how sensitive you are to competition overall. So it's the idea that you can produce way more offspring, which is the demographic part, but weighting that by how sensitive each of those offspring are to competition. So there's basically two ways to win. And in the actual paper, we've decomposed each of those two components to look at the evolutionary signals in each of them. Like, to what degree is it a demographic, like purely based on the intrinsic growth rates, or to what degree is it the sensitivity to competition? You can decompose them as well. Does that make sense? It's two ways to win.
I would call fitness the rate of growth, so the ratio between the next generation and the current generation, because whatever happening, the population which has this kind of fitness higher, then it will grows relative to the another one. This is the kind of general concept of fitness, but many people call the lambda as a fitness, which is the fitness without the ability, without taking into account competition. Okay, so maybe I'm not fast enough to think about it. Okay.
Yeah, I mean, you're right. I think what you're describing is you're kind of like, okay, if something's growing more in a generation, that means that it has higher fitness. This is actually, so that's kind of what would underlie the invasion growth rates, I'd say, which we're not invasion growth rates, but just growth rates overall. But the important thing is these stabilizing differences and the competitive ability differences, they actually both together affect population growth rates, but we can decompose them. And so there's a component of your population growth that is due to using those different resources and those stabilizing differences, and a component that's due to the competitive ability differences. And so if we're just looking at like overall population growth over time, we're kind of like looking at each of them together as opposed to being able to disentangle and partition their relative influences, if that makes sense. So I understand absolutely where you're coming from, and yeah, I think it helps to rename this away from a fitness difference, which is where you might think it's the population growth rates relatively over time, and instead just decomposing, like assuming they're actually competitively identical, how in what resources they're using, what's the asymmetry, and how good those competitors are for those resources. Does that help?
I think so. I speak Geza's language here. So the thing is, what this, and I can show you Geza later how it's derived in general. But the point is that in the chess on oeuvre of thinking about this, this quantity is a scaled measure of who would win the competition or how much faster one species would grow than the other. It's the ratio of the two discrete growth rates if they were occupying the exact, if they were regulated in exactly the same way. So if we match them up so that there's no distinction in regulation between them, then would be the ratios of the growth rates.
Yuri, just a very short remark that I think that Geza problem is that what is the mechanistic background making difference between the stabilizing and the competitive ability difference in terms of individual traits. So that's a problem. That why you make this type of division of fitness, partition of fitness to these two components, whether there is enough mechanistic explanation for that.
I just drew something really terrible, but this is one way that I like to think about it. So this is like a resource spectrum, and these are the curves of use, right? This is kind of what you'd get if it was purely just stabilizing difference or niche difference. Oh, this is a case actually where I didn't draw this enough. These are identical, completely identical. A case where they have the same niche, but one's just better at it. There's this kind of scenario, perfectly overlapping different heights. And then this one is kind of like both at the same time. So you can kind of see that they can be partitioned in trait. That you could imagine this being like a trait. It's like your consumption of whatever it is, like nitrogen or something.
It's always seed sizes of the birds, right?
Yeah, I don't know what it is for plants though. So
root depth.
Yeah. Hey.
But you can do the same with the other partition when you just get the intrinsic rate of increase and all the other stuff in the equations, and which fits these drawings as well.
And this is oversimplified. There's actually a lot of different ways that you could achieve different competitive abilities and things, but yeah. Simplification, but useful.
I would say a short answer to that is, of course, these equations that you see are completely phenomenological because the model is phenomenological. But in certain situations, there's a possibility to connect it to mechanisms.
Yeah.
For example, with that mechanism. Sorry, Liz, I do see that you wanted to say something else as well. However, I will ask Nadav, your hand was up for a while. I don't know if it technically still is and you wanted to say something.
Not really. I mean, just that I enjoyed the talk very much.
Yes.
I had a question, but I decided not to.
Are you sure? We're here.
Well, okay. If you press me,
please go ahead.
In the first part of your talk, what I gathered from it is that environment affect species composition via competition. It's not abiotic factors that sets a range limit of species. It comes from competition. The abiotic factor makes one species fitter here and one species fitter there.
Yeah.
And now in the second part, you had a series of experiments and you showed that if species are close by phylogenetically, then they are playing more or less a neutral game. So they have full niche overlap and zero fitness differences, approximately. And if they are, otherwise, if they are farther away, they have very complete niche segregation, so they do not compete anymore. So are the species, so I mean, I wonder, I mean, the impression from the first part is completely different from the impression from the second part. I mean, maybe it's different types of species. I'm not, of course, familiar with, but if it, I mean, what exactly are the relationship between the two?
Actually, that's a really great question, and I have a very specific answer for that. They're actually here's a secret. They're actually the exact same data set, and
okay.
what it was is, and they actually are compatible. So I'll explain why. The original, so actually the evolutionary experiment paper came first, and that was the initial goal of it. And so there's this idea in the phylogenetic community assembly literature that if the environment's really competitive, it should select for those differences in distantly related species. If the environment's really stressful, abiotically stressful, that it should select for similarity, species that have similar traits to contend with that stress. So that experiment was trying to get at both of those two things at once. And so there actually are wet and dry treatments in the phylogenetic distance stuff. And actually initially we had this whole expectation of how the answer will depend on the soil moisture environment. Yeah, it turns out overall, I think actually a big difference, I'd say, is that the filtering experiment is kind of dealing with where individual species and species pairs are falling relative to a persistence threshold, whereas the other experiment is instead looking across species pairs and trying to understand, phylogenetic distance as the predictor of those species pairs. And so each pair is modified by the abiotic environment. We know that from the filtering, wet-dry environment thing, but not in a way that distorts the phylogenetic relationships. And so soil moisture is actually in that model. And originally, again, we expected this like really interesting interaction that didn't end up happening. And part of that is, again, in the greenhouse, conditions are pretty good, and it does affect the outcome of competition. So individual pairs move around on the phylogenetics plot, for example, but not in a way that distorts the phylogenetic relationships. So they actually are, it's funny, they're because it's actually the same experiment, they are entirely compatible, yes.
Okay, thanks.
Thank you so much. Good to know. Liz, you did have your hand up for something else. I don't know if you still wanted to ask about that.
Oh, I had my, because of the former problem, that how you divide, how you partition fitness, and what is the mechanistic basis of that in terms of individual traits and regulating factors. That's all, and it was discussed.
Okay. Are there any other questions?
These are great questions.
I have one last question, which is this effect that you mentioned of because we're not measuring densities, we're measuring individuals very often and fitting these models, and therefore we get this spurious correlation between lambda and alpha II, or self alpha and growth. How many studies have it wrong? In particular, I'm thinking of those studies which have measured exorbitantly strong self effects compared to non-self effects. By that I mean, in a system with 4 species, I'm trying to recall, it was 100-fold difference, like 100-fold stronger diagonal entries in the matrix than off-diagonal ones, which even for those who believe in independent regulation might sound a bit exorbitant. Are those influenced by these methodological artifacts? Potentially.
It's possible. So what was happening? Yeah, I mean, it's hard to know. I think, when I do my experiments, they're usually in like a neighborhood of like this size. So it's possible having that one individual has a much larger effect than it might have for somebody who does like a larger experiment. But I wouldn't say that the pot size that I have is like atypical, so that, that might not be the issue. But yeah, I mean, we won't know unless we look. Actually, once we figured this out, we were kind of digging into the literature, actually, because we wrote this in our paper and someone reviewed it and they were like, why would you ever modify the Beverton-Holt model? And we're like, oh, we need to, or else this thing happens. But in addressing that reviewer comment, we actually found a few other people who actually identified the same problem and had modified their equations equivalently, which is great. It makes me feel really good. So yeah, it's hard to know what the impact is unless someone goes through their fitting and asks if it holds up.
I guess. Yes. Thanks so much. So are there
one
any
other
other questions?
Oh, no. Okay.
I'm sorry?
Can I say one more thing that I just remembered about that? One other solution. So before I knew what the problem was, so we knew there was this problem with the correlated posteriors, but we didn't know what the problem was. And so the solution we actually had from that was, because I do Bayesian model fitting, as I just said, a very informative prior on lambda, and that helped it constrain things. And lambda was because we had seed outputs on singular individuals. And so that was how we dealt with it originally. And now that we figured out exactly why that was happening, it's now we don't have to do any kind of constraining. It just fits properly on its own.
Meaning with a non-informative prior.
Exactly, completely, yeah, uniform. Yeah.
Sounds great.
Could I ask a question following up on that?
Oh yeah, sure.
So when you mentioned you had that correlation, was it like you had a sort of degree of freedom problem where you could sort of pick one parameter value and the other one you could get an equally good fit, or was it just that they were sort of a little bit correlated? Like, was this like an identifiability problem, or was it just a little bit correlated?
The former. It was an identifiability problem for sure. The thing was like this. It was like,
yep.
Yeah.
Yeah. Gotcha. Okay. Cool. Good to know. Thanks.
Thank you so much. Any more questions? If not, then thank you so much, Rachel, again, for the great talk and the great discussion. We will be back in 2 weeks and we'll be continuing. So we hope to see you then. We'll be in touch through email. So thank you again for your attention.
So the next lecture is by Bob Holt, and his title is COVID-19 meets Hutchinsonian-ish.
Thank you.
I absolutely don't know what it means, but it's title.
We'll find out from Bob in 2 weeks. So yeah, for that, thank you again for your attention and hope to see you there at Bob's talk in 2 weeks. Bye, everyone.
Thank you so much. Thanks for coming.
Thank you for coming. Bye.